There are different protecting groups for each amino acids. You have to select a perfect amino acid in order to achieve the good purity, otherwise it might not work. If you are really good and if you are solving any problem, the government will fund you to start your own company.
So for listeners outside the lab, how would you explain what a peptide chemist does day to day?
It's not that the first product itself will go and will be massive hit. Obviously there will be step by steps. You need a lot of patience, lot of experiments you have to run, sacrifice time you have to give.
So how has moving from India to Germany has shaped your career and research outlook?
Germans are really nobody is going to work on holidays. Yeah, work-life balance is amazing in Germany.
So today at Pharma Now we have Dr. Manoj Muthyala. Dr. Muthyala is an organic and peptide chemist with a PhD and research experience at Freie Universität Berlin. Author of 25 plus publications, his work focuses on sustainable peptide synthesis, green chemistry, and bioconjunction. He represents the next wave of peptide and complex therapeutic innovation with insights bridging India's talent and Europe research culture. So welcome Mr. Manoj.
Thank you. I should correct, it's bioconjugation.
Bioconjugation.
Yeah. And I did my postdoc in Freie University. I did my PhD in Pilani.
Pilani.
Yes,
yeah.
yes.
So, Freie University, can you tell me about more about
Yeah,
that?
after my PhD, I was looking for a postdoc position. Like I would suggest if somebody is looking for a postdoc, just writing emails, nowadays it won't work. So you have to attend a conference.
Oh.
You have to attend a conference and explain your research ideas to which you are interested in.
Okay.
To the professors. So that if they like your idea, they might ask you to write a proposal, and then that's how it worked for me.
Okay.
So I wrote hundreds of mails. It never worked for me. But when I went for a conference, I met professors in the same field, and then I tell them my ideas, how I'm going to work, which related to their field as well, and then I got an offer. So then I moved to our postdoctoral study. I went to Freie Universität Berlin. I worked under Professor Rainer Haag. He's working on a polymer chemistry, drug delivery. Though my PhD was small molecules, ionic liquids. So he was interested in how ionic liquids, polymer ionic liquids used for excipients in proteins. So that's what, so we had some ideas. I proposed some ideas and he liked it, and that's how I ended up in Freie University.
Oh, it's like a whole lot of process.
Getting a postdoc is like now it's a whole lot of process because a lot of PhDs are coming from India, and it's PhD is the highest degree.
Yeah.
It's a postdoctoral research is an research experience. Why we do it? It's getting more exposure, getting to know other fields. If you visit other country labs, then you will come to know how the research is going on. Yeah, and then I would say it became a normal thing to do postdoc after PhD. And it's not a mandatory thing, but it is becoming more quite normal.
So you have to pitch yourself.
Yes.
But it's for like only for German universities or for all universities?
All you can go to US. Like first preference would be US because they have a lot of funds, lot of research grants, and then if some people are not liking the situation, like current situation in the US, then Europe is also emerging. The Germany has lot of fund for the research. And this is a multidisciplinary research. That's good thing. It's multidisciplinary. So you'll come to know. So what if you have synthesized something? You will keep in mind before synthesizing that, like what it is useful for. It's a multidisciplinary field. Like then, you will get more exposure.
So, what inspired you to pursue peptide or organic chemistry as your research path?
So as I told you, I'm a small molecule organic chemist during my PhD. I did my PhD in Pilani under Professor Anil Kumar. We worked on green chemistry, which is ionic liquid supported reagents. That means you can reuse the catalyst.
Okay.
So to minimize the waste. So it's a green chemistry principle. So I am always interested in organic chemistry from my bachelor onwards. I know that I want to do my PhD in organic chemistry. So after finishing my PhD, then I was looking for postdoctoral position in the same fields, small molecules. Then I realized that there is a lot of applications in other fields as well. Like you can combine these two fields, polymers and small molecules in drug delivery, or you can use excipients in protein stability studies. So there is a multidisciplinary will give you more exposure. That's the reason I took. I spoke with the professor when he came for here ISCBC conference, I guess, and then he's working on polymers. So I am working on ionic liquids. I said we combine these two and how it can be useful. That's what.
Okay. So right from your graduation, you are into.
I am into organic chemistry. I know I want to do PhD in organic chemistry because I'm interested in chemistry as well.
Okay. So how has moving from India to Germany has shaped your outlook?
I would say during my PhD I was mainly focused green chemistry, synthesis, and how it applications, like how it can reduce the waste generation, how it can minimize. And my main focus was that. Once I spoke with the professor, once I read his articles, he was more into drug delivery, nanoparticles and everything. Then when I went there after taking the postdoctoral, like I visited 6 months initially as a guest researcher. He offered me 6 months guest researcher, sorry. And then I was there for 6 months. Then our work was going good. Then he offered me a postdoctoral position over there. Coming to Germany, like culture is little different compared to India. So here we respect our teachers. When our professor comes, we stand up. It's a funny incident happened during my first day. There is a conference going on, like group, this big group, and then the professor came. I just stood up. So everybody, all other persons were sitting. They looked at me. So why he's standing up? I said, okay, if professor is coming, we should stand up. Then no, and they are calling by his name, Professor Haag. Professor Haag, which is unimaginable for me. Like that's a cultural shock. And if professors come to you to see you in your lab, no need to stand, and you can sit. If he stands, also you can sit and you can explain. If he asks you to, do you have some time? You can even say, no, I am busy. I'm busy in doing. I can come to you later. That was a little cultural shock. Because I'm not saying ours is wrong, this is right. Ours is like we have Gurudeva Bhava. That's we respect, so we cannot say no to our teacher, and we respect our teacher. They also respect, but they can say no. They can say if they are busy, they say no. They won't they have their way of dealing as well. Their own way of dealing. I cannot say, I don't say that's disrespect, that's their culture. It's our culture. And it's a cultural shock for me initially.
So even the professor is okay with that?
Yeah, professor is okay with that. He used to ask us if I call him sir. No, don't call me sir. Call me by my name. Professor Haag or Dr. Reiner. So if he used to ask us, just call me Professor Haag. Reiner. If you are close to someone, you can call them by first name. If you are meeting for them first time, it's always better to call them by family name, second name. So if you are calling them by first name, that means you are really close to them in Germany. So everybody calls me Muthyala there rather than Manoj. It's like here everybody calls me Manoj. So no, they call by your family name. That's also a cultural shock.
Oh wow. Wonderful. Okay, so for listeners outside the lab, how would you explain what a peptide chemist does day to day?
So for example, everybody has, like, our body is made up of proteins,
Yeah.
everything. If it is 40 amino acid, considered as a peptide. Bigger than that one is considered proteins. So what we do is we synthesize peptides. We are a kind of, our company, what we do is we have a peptide synthesizers.
Okay.
So now peptide synthesis is completely automated. So what you have to do is, earlier one has to do manually. Some people does it manually now also, but most of them use synthesizers. So what we do is, synthesizer does is, you have to just measure how much amount you need based on the scale. For example, if you are synthesizing 100 micromole scale, how much you have to weigh, you just weigh, and you just give the sequence. See, there are 20 natural amino acids in the peptide field. There are unnatural amino acids as well, but natural amino acids are 20.
Okay.
So these 20 amino acids, and our proteins are made up of these 20 amino acids. So it changes the position. So you get n number of variations, like
in the synthesizer.
Since, like, not in synthesizing, our body as well. Variations. When the position changes, you get a different peptide.
Oh.
So there is n number of possibilities. So with only 20 amino acids, there is a huge number of possibilities and something. Our body has, it's proteins. Then what we do at our lab is we synthesize peptides.
Okay.
And we purify them. So purification is a bottleneck process. We have a gold standard for purification, what is called reverse phase HPLC. So this is purify. This is an instrument. We load that with our peptide with the column after completing the synthesis. It purifies.
Okay.
But what our technology does, we call as Peptide Easy Clean technology, PurPep. It's a chemical method. So what it does is, it's a catching a fish, kind of. You throw the net and fish out. So that means our neck is this one, a linker. So we throw it, and then it only couples to the full length peptide, and then we fish it out. It's kind of doing a funny way of saying fishing out. This is a purification part, once the peptide synthesis is finished. Yeah. So we do synthesis, then we purify, and we do deliver to the customers. Yeah, that's it, what we do.
Okay. So, and what technology do you use, to which is getting very useful in the industry, especially for peptides?
So as I told you, like, completely automated now. Peptide industry is completely automated. If you are going for the working in the research lab, pilot scales a little different. There you have reactors. Everything reacts. If you are working in R&D, there is a, based on your scale, there is a different reactors are there. For example, if you are working with 100 micromole scale, we PurPep chorus, what we deliver, what we ask to use the customers. If you are going for higher scales, we have Sonata instrument. In the middle, we have one more instrument. And then, based on the scale, we suggest this one. And then you have to, as I told you, we enter the sequence, we weigh the amino acids based on the sequence, and then it synthesizes, and then we purifies. But it's not that easy, because nowadays peptide drugs are becoming more complicated. Earlier, it's getting more bigger and bigger. It's 30 amino acids, and then side chain modifications and cyclic peptides. So you have to develop new methods. It doesn't mean that only synthesizers will do everything. So you have to select a protecting group in order to do chemoselective reactions, in order to form a disulfide bond, cyclization, fatty acid. So it's a lot more chemistry, So peptide synthesizers can synthesize based on your expertise.
Okay.
Based on your expertise, you have to know which amino acid you have to choose, because there are, though it is a 20 amino acids, like natural, unnatural, there are a lot. There are different protecting groups for each amino acids. You have to select a perfect amino acid in order to achieve the good purity. Otherwise, it might not work. You might not end up. You might end up with less yield or something. So how peptide chemistry works is you have a resin, and let's imagine like maybe take a string. Let's imagine a string with a ball something. So one amino acid come and attack here, and then second amino acid, like chain grows like this, like that. Why on solid phase? Why not we can do solution phase like small chemistry? What happens is in the solid phase chemistry, when you couple the next amino acid, excess you can easily wash away. Excess easily. So the purification, the synthesis become easier. If you do in solution phase, as it has carboxylic group and amine group, it can like a snake biting its tail. So you will end up with huge number of impurities. The solid phase will reduce the impurities.
And you can just wash it away.
Well, you can wash away impurities, though it is a solid phase. Compared to solution phase, bigger peptides, always solid phase is preferable, 100% preferable. Even solid phase, the crude purity will be lesser, then you have to go for purification like HPLC or our purification technology, which is called PEC.
Okay.
Peptide Easy Clean technology.
So are you saying like technology is useful, but also experience is needed? Experience matters.
Yes, experience matter as well. So you have to know the chemistry, organic chemistry, what are the protecting groups you have to use, what are the, you have to know how to, which reagent you have to use. So you should be a master in that like field like you cannot simply go, you cannot design new thing. If you are a technician, for example, technicians also work in our, we just give them sequence, they just work, they just, but if you have to design, if some problem comes, you have to choose, then expertise always needed.
Okay. So can you share how green chemistry innovations like DMF-free synthesis are changing peptide drug development?
That's a really good question, because we have published a paper recently. So EMA, European Medical Agency, has restricted using DMF in a larger scale. So if you are purchasing DMF, you have to sign a lot of documents and send them that, and it's carcinogenic as well.
Okay.
So it's dangerous. So you cannot inhale much. Like there is a limited. So we work in the hood, I think. So that so we came up with, like, lot of people are working on it. So we are not only one. So recently we got published a paper completely eradicating, removing the DMF and using alternative solvents, green solvents such as mixtures, DMSO ethyl acetate mixture, DMSO butyl acetate mixture. So this actually, it's working quite good. Now we have implemented in our synthesizers. Even in our purification, we completely eradicated DMF.
Wow, wonderful.
So
Okay. So which emerging analytical or synthetic tools are making the biggest difference in biologics today?
So I cannot say like all biological field, but I'm not expert in those fields. I can say that in our field, peptide field, some instruments are necessary in order to work. So like HPLC is mandatory and mass spectroscopy is mandatory. NMR, I say sometimes is helpful as well. NMR, nuclear magnetic resonance spectroscopy. So you have to come up with new techniques as well. So in order to and there are some techniques, some chemical techniques as well, some as chloroanil test. These are normal, like these are there from last maybe 60-70 years.
Okay.
So this is old thing. So now new technologies, I would say ours is also new technology in the purification. Peptide Easy Clean technology is a new technology, but it's not analytical technique. It's a chromatographic kind of a chromatographic technique.
Okay.
Apart from that, there is a need, but currently I am not that aware of any new technology. And automation. Automation is coming in every field.
Yeah.
For example, in our field as well, as I told you, synthesis is automated long back, and now this purification also we automated it now. So just like it makes job easier rather than doing it manually, sitting there adding reagents. If you develop automate it, instrument does it. Though you have to use your brain, everything, but manual labor has automation will help you in that case. So we have done in automation also our technology. It's helping us.
So we can save our efforts over there.
Yes. It's a manual labor will decrease as well in automation.
Use our efforts in innovating.
Innovating, yeah. That's what I say. What our technology also says. Funny say, we take care about your synthesis and purification. You do main innovative things. You design your molecules. You focus more there and give it to the other work to our technology. We do it for you.
Great.
Yeah.
Okay. So what difference do you notice in collaboration culture between European and Indian research environments?
Yeah, that's a good question. As I told you, when I worked in PhD, I was only working like alone. Alone means in my group.
Okay.
That's it. So in Europe and Germany, what I say, like it's mere application oriented. So if you are synthesizing a molecule, so what it is useful for, what application it is. If it is, it's hard to synthesize, doesn't matter. So what it is useful for. So more application, like there is a program called SFB 765, which I am the grant was coming for my postdoctoral research. You can imagine there are 40 professors in that group. So they all have collaborations. So 40 professors in one SFB 765 program, if I'm not mistaken. I don't know exact number, but there are a lot of professors as well. So what they come with different ideas. For example, organic chemistry is one professor is organic chemistry there, another professor is polymer. He says, why can't I immobilize my catalyst on your polymers chemistry and use a multivalency way. Other one has a peptide which can be showed antimicrobial activity, but efficacy is low. Our peptides can be easily degraded in the body. So delivering them, how you deliver. So then comes a polymer guy. He has a good delivery system to deliver the drug without degrading. So that's how I came to know that. So just not only like how good your molecule is doesn't matter. You need collaborations to finalize as a product. So there, and the other good thing is in Germany is if you are really good and if you are solving any problem, the government will fund you to start your own company. That's what our startup started from. My founder, he's a PhD guy.
Okay.
This is his PhD work. This is a PhD work. He started a company. So there is a lot of programs. He started his funding, though then company, government supported him. Initial phase, even they support for giving you space, instruments, everything. And then when you grow, then you can go out of the, like after 3 or 4 years, you can start your own. This one you can start. If you fail, no need to pay anything back.
Oh,
It's like yeah. So you are not keeping any your own money into it. So then you can take risks. So that's the key difference. Now also there, I think in India also the startup culture is starting right now. I think in IT it's huge, I guess. But in chemistry, in the science field, what happens? You just don't. You also need with your brain. You also need infrastructure.
correct.
Infrastructure needs money. Like if you want to buy a simple synthesizer, it costs you a lot of money, and HPLC costs you a lot of money. What happens in Germany, like when you have a new idea, you pitch, then they will go through your pitch and then say whether they think whether it is possible or not. They will fund you. They will even give you salary. They stand beside you. They encourage you. If you fail also, it's okay. It's okay. There is no end of the world. You can go and start work in a company. So that's the main difference I found. But now I think in India also, the startup culture is like huge.
It's a very wonderful culture where country is really helping out to innovate.
Innovators.
And because when you innovate, new things comes in the market, and then you're like, after all, you're saving lives.
Yes.
At the end of the day, patient matters.
Yes, patient matters.
So,
I know few of Indians also who started their company there. One of my friend, he did PhD. He has nice ideas, and he pitched there, and then they liked the idea. And he got really huge fund, and he's even doing now animal studies with his peptides. This is his wound healing. He's working on wound healing peptides. Wow. So there are a lot of opportunities if somebody interested in. If you have nice ideas, if you have patents, even more. There is a chance of getting fund is more if you have patents, and if your idea is novel.
Yes.
And there is encouragement. There is a startup culture. They will be inviting you. There will be like, if your idea is great, they will give you prize like to encourage you.
And now with the help of technology also, so it's getting more easier. We just have to have that idea.
Yes.
And with the use of technology, we can leverage the both, and we can make the best use of it.
That's true. I don't know about like the other fields, but science fields, idea, yes, you should have an idea, but expertise also matters. Just an idea cannot do things. That you should have good expertise in that field in order to bring something out. And it's not like the first product itself will go and will be massive hit, and then no. Obviously, there will be step by steps. First product, if you come to the market, then only you will come to know what are the pros and cons. Yes. Then cons will be, you will come into know cons. Then you will go and modify, you tweak, and then one day final maybe ideal product will come out. But you think that okay, this is the final product, it's going to work. It doesn't work like that in science, especially. You have to keep on tweaking. You have to talk with your customers. You tweak. It's a step-by-step process. You need a lot of patience. You need a lot of patience. Money-wise, they take care, but you need a lot of patience, lot of experiments you have to run, and little sacrifice. Time, you have to give time and efforts. I know like how my boss works. So he gives a lot of energy. But the work-life balance is really good.
Work-life balance?
It's really, really good.
Can you talk more about that?
Yeah, that's like Saturday, Sunday, your boss won't call you at all. You can completely relax. After 5, your boss won't call you. And there is a flexibility. If you say, look, my child is, I have 5 years son. He goes to the kindergarten, like Kita, the play school. If I say, look, I have to drop him at 9 o'clock, so I will come to the work 10 o'clock and work 6:30, 7 late. Or if my wife drops him, I go at 7 o'clock, I come at 3:30, it's 8 hours, work for 8 hours, and then it's a flexible. You can change anytime, like you can shift from 7 to 7, anytime you can choose. If we are like, they prefer like not after 8 don't work. So in between, starting from 7 or 6.
You just have to work 8 hours.
8 hours. But on the other hand, you have to do everything by your own, right? You have to take care. There is no help from the parents. But work-life balance, I say, and we get 30 days holidays without weekends.
Okay.
Apart from the weekends, we get 30 days holidays. It's a funny way saying, I spend more time with my parents right now in Germany when I stay in Germany rather than in India.
Oh.
Because I completely come here 30 days and I stay there with them and I spend more time. Though it's like, yeah, in India, if you can go only on vacations, which is short vacations, and yeah, 30 days, amazing, I say. And Germans are really, really strict about their holidays. Nobody is going to work on holidays. These 30 days, okay, I work one day, I take extra amount. No, Germans, they're really strict about their holidays. They plan their holidays 1 year before. If you ask a German what you are doing next year for the holidays, they have a plan. They have a plan this year. They sort it out. They know they book flight tickets. They have they are the timetable. They're punctual. If you come late, it's okay. You have to inform them beginning. As I said, my kid goes to teachers. In these days, I'll come at 10 o'clock. It's okay. So coming late is they consider as like you're not showing politeness.
Okay.
If you don't inform them. Communication matters. And then you say you deserve your holidays. You have to take. Don't work on holidays.
No.
If you work for extra hours also, they say you have earned it. You have to take holidays. That's it. I feel that they are less stressed. They are less stressed and more focused. 8 hours. Give it 8 hours. Completely focused. I never you cannot say 100%. I saw most of the Germans, they work 8 hours really focused. And
maximum productivity.
Maximum productivity. After 8 hours, shut, evening go. They say Feierabend. That means celebrate your evening, like free evening, Feierabend. They say evening, whenever we go, Feierabend. Celebrate your evening or something. Every day, they say. Yeah, it's good. It's something we have to take from them. It's okay.
So, what are your plans on holiday?
Good question. The thing is, they divide holidays so that every 3 months they take some holidays to not overload or something. I also earlier I used to take all holidays, collect all holidays, and come in India. Then I realized that it's taking so much pressure on me. So what I also dividing some holidays like 1 week every 3 months or 10 days. So if you combine the weekends and if you take some holidays also there for other than these 30 holidays, public holidays will be there. So we call them bridge holidays, as you might know. So Thursday is working. If Thursday is holiday, Friday is working.
Okay.
Then we take that holiday. They will be cut from our 30 days holiday. And we travel. Somewhere. So yeah. I used to take 30 days initially at the same time. Now I'm dividing. But obviously I miss my parents, so I come to India and then I travel with them here in India.
But there's no problem in dependency, if you are out and you cannot, you are on leave or you cannot work at that time.
Yeah.
So if any of your subordinate or your colleague needs you, wants you.
Yeah, that's a really nice question. Yeah. So here you'll get a call, right? So the phone. That's what. Your record maintenance is 100 exact. What you have done is you have to write everything. Everything they take really serious in the manual maintaining. So initial days I struggled. So why they are asking? This is simple. Why should I write it? No.
Write it.
You have to write each and every point. So that if a person who doesn't know anything, which is not from that field also, if you read, he can able to reproduce it.
Got it, yeah.
So it's reproducibility. So maintaining record for them is the, so it doesn't matter if you are a very good chemist, if you finish the projects, they appreciate you. But if you don't maintain the record, no, they say, this is not good. This is not at all good. That's the reason. That's a really good question you asked. In that case, no need to disturb you, right? No need to bother you in those holidays. You can go for holidays. Yeah, it's on the other hand, 1 or 2 calls is okay if something happens, but it's not keep on bombarding calls with your holidays. They say no call anymore. Yes.
In case of emergency.
In case of emergency, you call them if they are okay. But yeah, But in small things, everything is recorded, 100% manuals are there, so you can just follow that. And as I told you, like it's 1 year before they know
That this
so
person will not be
will not be here. So
Yeah.
you know prior, like now my boss knows when I will take holidays, so he's prepared.
Okay.
I know when my subordinate, like my other people, employees, other workers will take holidays, so I know. So I plan, I won't give them projects that time,
Okay.
because you have to try to finish them. If it doesn't finish, then give it to, don't work till last day, stop it and give it to other person, explain him so that he can continue. That's how it works.
That's really amazing. Like everything is planned, everything is recorded.
Everything is recorded. I say, like, even though some problems can come, right? Like even though they are planned, they think, if you imagine just completely unplanned, like there is no plan, there is no record, so lot of problems will come. That's the Germany is the law of rules. They say rules. Somebody, if you are initially going to the German, they say, look, they are little, people will say that, oh, they are harsh. I feel they are not harsh. If you follow the rules, they are friendly. If you don't follow the rules, even in the traffic, they don't like you. They will shout at you. Some people shout at you if you have to follow the rules in the traffic from top. So for example, for kindergarten, my kid, if I cross the red light, he would stop me. Dad, that's not, Papa, that's not right. You should stop on the red lights, because in the kindergarten they teach you traffic rules. So the rules are important for them. If you violate the rules, they are not happy. It's a cultural difference, yeah. For us, okay, sometimes, okay, it's a diet, we can cross it. Yeah, the rules.
But this is the way they live. This is their normal.
No, normal.
I think one more thing, we should take it like the recording thing which you said. Everything should be recorded.
Everything should be recorded, written. Now it's like it's IT.
Excel
Everything
sheets,
is IT, so you have to monitor it. But we do maintain our manual records as well.
Amazing.
So it's mandatory there. I think it's even in our pharma field also, it's mandatory. It's like but in R&D also they take it little more serious. But here also, I think in pharma, everybody take it serious. Everybody take it serious.
Great. Okay. So what skills should young scientists develop if they want to thrive in peptide or biotech research roles?
Yeah, first of all, basics. I say the basic knowledge. So I'm a small molecule chemist, organic chemist. Then I moved to polymer. Then I moved to peptide chemistry right now. And we work in bioconjugation as well. So the thing is, chemistry, you should have really good basics. And then you can apply this technology, apply your basics anywhere. So like small molecules, then come to polymer. The properties will be different, but the science behind them is similar.
Okay.
Science behind them. So you have to get an experience, handling experience, in order to work with them, because all is not similar. But if you know the basics, you can thrive anywhere. So I say fundamentals are really important. So learn fundamentals and learn handling and safety measurements. You should be really safe when you are handling those toxic environments. There I forgot to mention it. They take safety really serious. They take safety really serious. And so for young scientists, I say, like, basics, knowledge, acquire knowledge, and then experience, work, acquire knowledge, and then you can thrive in any field. Any field means any biotech, and know the new technologies as well. Read more publications. Reading publication, you have to update yourself. Keep on reading publications, like reading a newspaper. Reading is, you have to read publications in your field. It's impossible to read all the publications. If you want focus in one field, read those publications, acquire knowledge, then that's it.
Okay. How do you see like automation and AI transforming peptide synthesis over the next 5 years?
Yeah, so as I already told you, we are automating our synthesizers and purification. So what it gives you is the data.
Okay.
So automation and then AI, if you have data, for example, then if you feed the data, then it can give you where if a new sequence comes, it can tell you possible failures, where the possible failures can come, where the possible problems might come. You can use this data AI so that you know where my problem comes and you are prepared for it. Then you can fine tune or something. Or if you don't have data, if you don't have AI, then it might be like you have to spend more time doing the repetitive things. If you have data, like, and AI is really good in giving us data, more pulling the data from all the sources, and you have to be cautious as well. Then yeah, this gonna help you to achieve your goals faster. Goals means molecules really faster. Yeah, it makes things smoother.
So AI just gives data, or it really gives simplified data?
It gives you data, and sometimes it simplifies data as well, but you have to be cautious whether it is 100% true or not, because it pulls the data. AI is like pulling the data from all the sources. It might be sometimes it's wrong as well. I was surprised to see when one time I gave the CAS number. CAS number is each chemical has a number. CAS number. I asked AI a CAS number, and that it keep on giving me the wrong chemical name. I said that this cannot be because it's pulling from the different sources.
Yeah.
I say language models, I feel it's really, really good. Language models, writing proposals, like you have to feed it, you have to give your ideas, then it can fine tune more. And the research level, maybe it has to grow more, grow really more. I'm not that expert, but based on my experience, I can say, yeah, language models, it's amazing. Searching engines are amazing if you want to pull some data, and merging from all other, like if you give topic, it can pull the data from different sources and give you, but you have to be cautious. You have to look whether it is pulling the data, right data or not. Yeah, use it and cautiously use it. Yeah, that's it.
Okay. So what's one project or paper you are especially proud of, and why?
So in my PhD, I'm proud of green chemistry papers, which we like some of the toxic reagents. We produce alternative green reagents, green catalysts, which gives me immense pleasure. When, for example, one sulfonyl reagent was explosive in nature. So we developed a reagent, ionic liquid supported sulfonyl azide, which is easy to handle, which is not explosive in nature. We have done DSC studies, and then it gives you that satisfaction that you have given something to the research. And then coming to the working to the company now, we are also like, I'm working in R&D. So you deal with real problems. Like if somebody asks you to synthesize, for example, now we are working a project, MRI agent. So it's a peptide based MRI agent. So it has a metal in it, gadolinium something. So it's now they are studying in the pig models. Then you see like, okay, this can be potential of going to some, maybe, I don't know, it might fail or in future or something, but it can be real application. You can see real application what you are doing in the real world. So this gives even more satisfaction, right? Like, okay, you are doing something useful. Yeah, that's I'm happy about. Yeah, this interdisciplinary research. If something comes out of your research, it might not be product, but if addresses some problems, that's good. For example, in this technology also, my boss, I said, like he addresses the issue of purification. He came up with the technology. So it's solving problems. It might not solve all problems, but it's solving some problems. It's use. it's real use. You can see in from your eyes.
Yeah.
That gives immense pleasure when something comes out of your research.
And it's a collaborative world, like
Yeah, in the universities collaborative world, yes. In universities, yes.
so your innovation can solve can solve small problems, but in a collaboration it's contributing to a big solution.
Yeah. It is. If you, that's what, if you are coming, like all expertise coming together,
Yes.
then I might not have expertise in one field. So he has this problem in that field, but my expertise can solve that problem. So if he can explain, if I can understand, if we come together, then we both can sit and solve that problem. So interdisciplinary is the way to go. I say, like, yeah, basic research helps you. Basic research will give you tools to solve the problem. Interdisciplinary will, you can see something coming out of that.
Okay. So can you play a quick rapid fire round?
Yes, I'll try. I'll try.
Okay. So one word that best describes you at work.
Enthusiastic.
Wow.
Yeah, like I want to try new things. Like if something works, I'll go happy in the evening. I'll go happy and I share with my wife as well. Enthusiasm, like trying new things.
Okay.
I think every R&D person is
Enthusiastic.
enthusiastic.
Are you a morning person or a night owl?
I love to be morning person. I hate night. Morning, yeah, morning person.
Innovation or compliance? What excites you more?
Innovation.
If not in pharma, which career would you have chosen?
So I love teaching.
Acha.
I love teaching. My father is a teacher, so I am sure that in one day I will go to teaching once I retire.
Oh.
I will. Teaching is my other passion.
Okay. Coffee or chai during long research hours?
Yeah, chai, but after going there, now coffee, because making chai is really tough. You have to boil everything. Coffee, just keeping a mission if something comes. So in India, obviously chai. In Germany, coffee.
Coffee. Difference.
Different, yeah, different.
Okay, Dr. Manoj, it was really nice having
you.
you on Pharma Now, and it was really great listening to you and your journey. It's really inspiring.
Thank you very much having me here as well.
Thank you.